Okay, this video is about, does inflammation and infection cause cancer? So first thing we got to make clear is, what's the difference between a prokaryote and a eukaryote? A prokaryote is like bacteria. Pro means pre, karyote means nut, like in Greek. So pre-nut means pre-nucleus, and that's a bacterial cell, a prokaryote. And it just has all its enzymes in the cytoplasm. It has a normal outer plasma membrane, and then it has an outer cell wall. It'll use that for its proton gradients. Okay, now here's a eukaryote. This is what humans are. We're made out of eukaryote cells. So this means true nut has a true nucleus. But I want to share with you that all the high school and college biology books, they almost always get this wrong. They'll say, well, a eukaryote cell has membrane-bound organelles. The main point of a eukaryote cell, and this is a big point, is it has mitochondria. That is the defining, the important feature of a eukaryote cell, mitochondria, because that's where all the energy comes from. The only reason multicellular life exists on Earth is because eukaryote cells have mitochondria. And where does that come from? You know, many years ago, somehow the world started out with no oxygen, anaerobic, and there were bacteria that were able to live in those conditions, and then oxygen started becoming available, and then they were processed by mitochondria. The mitochondrial bacteria was ingested, so to speak, or it got inside of a anaerobic bacteria and then provided a symbiotic, meaning a mutually beneficial relationship where it would make energy for the cell that would enclose it and sort of protect it and help it to get food. And then eukaryotes have just took off from there. They sometimes call it the LUCA, last universal common ancestor. The important point to make is the defining feature of a eukaryote is mitochondria, and nothing exciting or high-level sophisticated is going to happen without mitochondria, because you need that energy to do sophisticated things. All right, and this is all about what's going on with cancer. So now we got to talk about the Warburg effect. Otto Warburg won the Nobel Prize in 1931. The guy was a German biochemistry genius. Some people think he was the best biochemist of the whole 1900s. There's, of course, a lot of other great ones. Okay, well, anyways, here's an anaerobic bacteria. Anaerobic bacteria takes glucose, runs it through glycolysis, makes it into pyruvate, then gets rid of it as lactate, and that causes lactic acid. ATE just means a deprotonated carboxylic acid. Carboxylic acids tend to be deprotonated in aqueous water solution. So this is the typical thing. Whenever you see ATE at the end of a word, that means carboxylic acid. All right. So anyways, that's what an anaerobic bacteria does. Now here's a normal human cell, a eukaryote. It's got a mitochondria, and these can make tons more energy from glucose. Typically, you run glycolysis, you get net 2 ATP. You run oxidative phosphorylation with the mitochondria, Krebs cycle, and electron transport and all that, you'll get anywhere from 30 to 38 ATPs per molecule of glucose. The reason it's a little variable is some of that energy from the proton gradient can be used for other things, to pump other chemicals, to generate heat. Okay, but overall you can get as many as 38 ATP from it. That's a lot. Okay, so anyways, then Krebs cycle basically harvests the electrons. We talked about this in my previous lecture on Krebs cycle, and it gets rid of CO2 as a waste product. So humans make CO2 as a waste product. All right, and now let's talk about a cancer cell. In a cancer cell, the mitochondria is damaged, meaning especially the electron transport, oxidative phosphorylation for making ATP, that's the damaged part. So what happens? And this is the key point of this whole talk. The human cell that has become cancerous now becomes like the anaerobic bacteria cell. This is a key point. All understanding of cancer necessitates that you get this point, okay? The damaged cancer cell no longer functions like a normal human cell. Its biochemistry now mimics an anaerobic bacteria. And some interesting things happen. First of all, its pyruvate is metabolized to lactate. And it'll pump that out of the cell, and it'll cause lactic acidosis around the cell. That lactic acidosis helps to give the cancer cell an advantage over the adjacent cells. This is part of the tumor milieu, meaning the surrounding extracellular matrix around the cancer cell. So an acidotic milieu favors cancer. And that's going to have effect on treatment decisions. A lactic acid pumped out causing an acidotic tumor milieu favors the cancer's growth over the adjacent cells. All right, now the inside of the mitochondria, in the matrix, we're not talking about electron transport oxphos. That's messed up because there's no oxygen, or there's a severe deficiency of oxygen. Okay, now the the Krebs cycle will go in reverse. And this is used now to make stuff. Instead of CO2 being expelled as a waste product, CO2 is taken in and incorporated to make more molecules. In addition, the most abundant amino acid is glutamine, and glutamine uptake into the cell will be increased. The cell will have increased transport channels for glucose, like glucose type 1 transport channels, and it'll have increased uptake receptors for glutamine. And this amino acid will be used to help generate intermediates for the reverse Krebs cycle. So the key point, though, is when you're only burning glucose to pyruvate through glycolysis, I have a previous lecture on glycolysis, you're only getting 2 ATP. That's hardly anything. That's at least 15 times less than what is obtained with oxidative phosphorylation on the inner mitochondrial membrane here. You know, the proton gradient, ATP synthase, harvests the hydrogen proton to then make ATP. So the point is, cancer cell has to burn tons and tons of glucose. It'll have lots of glucose receptors on here in order to generate enough energy to survive and to grow. And now we're going to talk about why does it do this? Cancer takes on a life cycle like an anaerobic bacteria. Bacteria will sit around dormant, and cancer can sit around dormant for years and years, perhaps decades. And then when everything it needs becomes available, all the growth components, it'll grow like crazy. That's what bacteria can do. Bacteria can replicate itself 72 times in one day. That's rather incredible. You know, every 20 minutes, a new bacteria can be made. So what's the rate limiting step for bacteria? The rate limiting step for bacteria is to replicate their DNA. And bacteria tend to have relatively small amounts of DNA. And that's because it's the rate limiting step. You got to make all those DNA base pairs, and that slows down replication. Whereas a human cell's got tons more DNA. So a bacterial cell, let's say it has 10,000 base pairs. It might have about 10 million, about the most it'll have. Whereas a human cell's got 3.3 billion, and you got to make a copy of each side of that chromosome. So you're talking, we have to make 6.6 billion base pairs of DNA. That's tons. They're made out of nucleotides, and that's tons of synthetic material to make that. So remember that. Cancer is very much resource dependent. All right, so once it has everything it needs to grow, it can grow super fast, exponentially fast. All right, then it'll hit a plateau where it starts to deplete itself of nutrient resources. Then it'll start to die. Because when you think about it, cancer is a little bit stupid. What I mean by stupid is it grows real fast, metastasizes everywhere, kills the patient. Cancer itself would be better off if it grew a little slower, didn't kill the patient, but it can't control itself. That's what cancer does. So we want to remember this, though. This is how cancer can be. An anaerobic bacteria can be dormant for a long time. They get what they need to grow, and they can grow really fast. That's why you don't want to give cancer anything. You want to deplete cancer of iron. You want to deplete it of methionine, leucine, lysine. Okay. Also, it can acquire, anaerobic bacteria can acquire DNA from adjacent bacteria for plasmids, for example, lateral gene transfer, through a conjugation pilus and horizontal gene transfer. So I just make that point because, cancer can also be kind of tricky in the sense that a person could receive, a chemotherapy or something, and you could deplete 99% of those cancer cells. The tumor shrinks dramatically on CAT scan. Everybody's happy. But if those cancer stem cells are still alive, they can be dormant for a while, and they can grow right back and now be resistant to that previous treatment and, lead to the patient dying. All right? And the reason we want to understand cancer is this is how you have the best chance to get the best results and to survive it. Okay, now the big question of this talk, does inflammation and infection cause cancer, and why? By the way, you always want to try to understand why when it's possible because it helps you to make more intelligent decisions. Okay, it's like when I train residents how to do procedures, I always ask them, why do you do this? Why do you do this? Why do you do this? And some people think I'm being a little annoying. Why do I do it? The reason is, I've done many thousands and thousands of medical procedures, just that you run into surprises. And if you understand why you do every single thing and what each piece of equipment is good for or not good for, you can just work around the problem, okay? And so that's what I'm trying to do, is to help a person who wants to survive cancer, is to understand it so you can see what the workarounds are and you can understand what cancer does and you can have a better idea of how to prevent it. So here's some things that cause inflammation. Asbestos, that's an ingested foreign body. It'll go in the lungs and there'll be an inflammatory reaction around it. Silicosis is kind of a similar concept. This is a little bit different. Moldy peanut butter will have aflatoxin, and that can lead to liver cancer. That's sort of a big thing from T. Colin Campbell's research. We'll briefly talk about hiatal hernia causing GERD, gastroesophageal reflux disease, Barrett's esophagus, esophageal cancer. And by the way, what causes hiatal hernia? Abdominal pressure syndrome. Like, Dennis Burkitt figured that out. Lack of dietary fiber, constipation, chronic straining at the stool, valsalva maneuver, increased abdominal pressure, leading to the stomach to pop into the chest. It can be small or it can be big. It can be half the stomach or the entire stomach. It leads to gastroesophageal reflux with the acid burning to get that heartburn-like pain, and that'll lead to a compensatory change in the esophageal lining cells called Barrett's mucosa, and that's predisposed to esophageal cancer. So we know that inflammation predisposes to cancer. We know that some infections predispose to cancer. Crohn's disease and ulcerative colitis are forms of inflammatory bowel disease, and those increase the risk of bowel cancer. Okay, now HPV, human papillomavirus, is associated with cervical cancer. It's also associated with nasopharyngeal cancer. That's thought to be like the one, remember Michael Douglas, the movie star? He's thought that that's what he got it from oral sex. Theoretically, that's what's thought to have happened. Epstein-Barr virus associated with Burkitt's lymphoma. EBV, that was a little bit overrated. So many people have it. That's kind of a topic for another day. Helicobacter pylori and gastric cancer. So there's increased incidence of Helicobacter pylori in persons who got gastric cancer. By the way, vegans are much less likely to have Helicobacter pylori. Okay, the treatment isn't just antibiotics. All right. Okay, hepatitis B virus, HBV, hepatitis C virus, HCV, increased incidence of liver cancer. I'm going to tie all this together. The goal of this talk is to make sense out of all this stuff. All right. HPV, we talked about that being associated with cervical cancer. Schistosoma, causing chronic inflammation of the urinary bladder, leading to bladder cancer. Bovine leukemia virus can potentially cause cancer. All right, so now Otto Warburg knew back in his day, like the 1920s, there were a lot of different things associated with causing cancer. But what Otto Warburg figured out, he's the PhD Nobel Prize winner from 1931, he figured out there's only one primary cause. Lots of secondary causes, but only one primary cause. So the real question is, how can we tie all this together to one cause? Here's what Otto Warburg says. The primary cause of cancer is the change from glucose metabolism with oxygen, like eukaryote cells, human cells, to anaerobic fermentation, to lactate, like an anaerobic bacteria. And that's my point, why I said cancer is transformation of a human cell to becoming like a bacterial cell. Trust me, if you get that, tons of things about cancer will become more clear to you. Cancer grows in oxygen-deprived tissue. All right, this is in red, and it's in large letters because this is like one of the most important points of this whole talk. Cancer grows in oxygen-deprived tissue. That's the Warburg effect, named after him. The W's are often pronounced like V's. The Warburg effect is cancer grows in oxygen-deprived tissue. Warburg showed if he deprived a cell of 35% of its oxygen, they would, many of the cells will die, but some of the cells will turn into cancer. So oxygen-deprived tissue will turn into cancer sometimes. So you don't want your tissue oxygen-deprived. Now, here is asbestos. So this is the trachea. This is the left mainstem bronchus. Some asbestos fiber is accidentally inhaled. The asbestos fiber comes into the lung, and it's a foreign body, so the immune system forms an inflammatory response to it. Inflammation starts out being a hot disease, so to speak, and then collagen, which is fibrous tissue, scar tissue, is laid down around it, fibrosis, and that walls off the foreign body to try to prevent it from causing more trouble. You get a little bit of edema. That means inflammation-related fluid around it. Now, the thing about this fibrosis is it's going to block access of oxygen to the cells that are trapped in there, the inflammatory response cells and the lung cells and the pleural cells. Pleural is just a lining of the lung trapped in there. So guess what? Some of them are going to die, but there's the potential that some of them will be transformed into cancer by the Warburg effect to become like anaerobic bacteria. and then they can grow and cause cancer. Asbestos is associated with increased risk of lung cancer and increased risk of cancer of the pleura. The pleura is the lining of the lung, and that tumor specifically is called mesothelioma. Now we're going to show, here's an example of what will happen in the liver. Here's a nice normal liver, healthy liver tissue doing the job of a liver. And then the person keeps eating too much high-fat food and they get non-alcohol fatty liver. We'll just call it fatty liver up here. The fatty liver can become inflamed, and that's NASH, non-alcohol steato, meaning fat, hepatitis, liver inflammation. And these little gray lines are increased collagen being laid down and fibrosis. And that'll progress till it's really bad, and then it becomes called cirrhosis. Cirrhosis is characterized by nodular bumps on the surface of the liver. And all this fibrous tissue is depleting the liver cells of oxygen, and guess what? You can get a cancer. Now, I realize with infections there's potentially genetic changes as well related to the infecting particle, but the point I'm making is Warburg had incredible insight back in the 1920s that lots of things can lead to ischemia of a cell with subsequent transformation of some of those cells to cancer. So remember that. And we can do a lot of things with our diet and our lifestyle to avoid having our cells be ischemic for prolonged amounts of time, especially like more than 48 hours. Number one, by avoiding diabetes and hypertension, by eating a low-fat diet and a low-sodium diet, we'll have much better oxygenation of our tissues. By avoiding routinely eating high-fat meals, we'll get better oxygenation of our tissues. Okay? All right, that's it for this talk. I'll go with more pathophysiology, cancer, and other talks and help you to develop a deep understanding of it.